So yeah, good afternoon already, everybody. Thanks a lot for joining us, and also thanks Eric and the Covaris team for the invitation to present today. So in fact, I'm a product manager at PacBio, looking after our DNA extraction solution, but also some other application, and it was very interesting for me to get more familiar with what we can do with FFPE. So yeah, as many of you may know, FFPE samples represent a huge opportunity for cancer genomics, but they have historically been challenging for long-read sequencing. So today, I will share with you how this workflow addresses those challenges and the performance that we have seen with some FFPE samples. So yeah, just checking. Most clinical tumor samples are stored as FFPE, and it represents an incredible resources because they are often linked to pathology information, treatment history, clinical outcomes. But the challenge is that the FFPE preservation introduce DNA damage, as Eric was explaining during the introduction, which has historically limited the use for long-read sequencing. So while the samples are very valuable, they are usually not used. And honestly, before being a product manager, I used to be an FAS working directly with customers. And when someone was asking me about, can I sequence FFPE sample with PacBio, I was like, no, I don't think it's a good idea. So yeah, I'm very happy today that we have something that looks much better. So then the second topic, of course, when we think about FFPE is cancer. So why using PacBio long read for cancer? So in fact, because the genome, the cancer genome are usually much more complex. So we are not only interested into a SNP, but also, of course, structural variant, repetitive region, and also it's important to understand whether the variant occur on the same haplotype or on different haplotype. So long-read sequencing is, of course, an ideal tool for that. Here you have an overview of what PacBio can do with cancer. But when we work with FFPE, we really focus on those area here highlighted with the box, meaning, of course, WGS, comprehensive variant detection, phasing, repetitive region, and structural variants. So I will continue with a short introduction to HiFi sequencing. Maybe I should ask in that room who is familiar with PacBio sequencing. I know someone on the first row. Oh, great. So yeah, that's good to see. For the ones that are not familiar, just a quick recap. So usually we start PacBio sequencing with high molecular weight DNA. So now you will think, what she's talking about, FFPE, high molecular weight DNA, but keep that in mind. Then we ligate our smart bell adapter at both extremities of the molecule. So like that, we create a circular molecule. And just before sequencing, we bind our polymerase here at one extremity of the molecule. Then we start sequencing on our instrument, could be Vega, Revio, depending the throughput, and we generate a very long polymerase read length that could be over 100 kb. And at the end of the sequencing run, we align all the sub-read to only keep one HiFi reads that is really high fidelity, because we correct the error with all the different subpaths. So it's how we can get into over 99.9% accuracy. So basically, to recap, HiFi reads are much longer than short read, but of course as accurate or sometimes even more than short read. And yeah, the main application, the main advantage for today for FFPE cancer project, I would say it's really comprehensive variant detection with, of course, SNP in DEL, but also larger variants and phasing. So now I will come back to the FFPE challenge, and yeah, as you mentioned, Eric, that's really start during the fixation, because the formaldehyde who preserve the tissue extremely well, it also damaged the DNA. So then it creates some protein DNA crosslink, and it's where we start to have issue when we do PacBio sequencing. So yeah, the result of this fixation is usually low DNA quality, and it's then difficult to use this sample for molecular biology workflow like library prep. So yeah, the question was really, can we do long-read sequencing with FFPE sample? And it's really what we are trying to address with our new workflow. So this is the workflow we present at AACR earlier this year, end of April. So it's fresh from the press, I would say. And we also have a tech note describing that. But basically, we start with the truXTRAC extraction protocol that was described earlier, and then we use different components of the PacBio workflow. So amplify the DNA that we get, and then some Kinnex PCR to generate artificially long molecule, and then we sequence that and finish with the analysis. So this is really the key innovation behind the workflow. So Covaris addressed one side of the challenge by recovering longer DNA fragments and improve the DNA quality from FFPE. And then PacBio addressed on the other side the input of FFPE. So for that we have the amplify to get more DNA, and then the Kinnex to do the concatenation and end up with a long read to start the sequencing. So really together both technology help overcome the key limitation associated with the FFPE derivative sample. So yeah, this is a quick look of the workflow, how it looks really in the lab. So for this study, we started with 2 to 4 FFPE tissue, 2 to 4 little scrolls, and we had the chance to work with kidney, uterine, and brain tumoral tissue. So we start with approximately 50 nanogram of input DNA, and it took us more or less 2 days from beginning, so from extraction until starting the sequencing. So really the key message here is that with this workflow, we were able to start sequencing FFPE. So then some sequencing performance, of course, the most interesting. So for all the sample, we were able to get more than 100 million of reads per sample. So I would explain a bit more the data that I'm showing here. You see the different samples that we tested, the tissue, the number of reads that we obtained, the yield, and the length. So this is what you get as the output of the sequencing run with the long DNA molecule that we create with the FFPE during the library prep with the Kinnex workflow. And then, of course, we, during the first step of the analysis, we cut back the long DNA molecule into the real FFPE sample that we use as input. And it's why you see here the real, I have a problem with my pointer, the real number of read. So let's say between 110 million up to 160. And then the read length, and it's normal to see a variation because this really depends on the length of the DNA that we are extracting with the Covaris solution. But with that length, you will see that it's already very interesting in terms of variant analysis. So then of course the next step, the variant detection. So we were able to detect, let me try to take this real, okay, no, it's okay. So we identified 11,000 structural variants per sample. This is usually what we typically see. Then over 5 million of SNPs, and 60% of the variants were phased. So just to give you an idea on the table, I think interesting to see the mean coverage that we get for those samples. So you see that it's varying from 12 to 34x coverage, and this is really linked again to the size of the DNA that was extracted. And yeah, to finish on the analysis, we also tried doing some phasing. So we decided to take HLA loci as an example, and we clearly see that when the samples are larger than 1 kb, in fact, it was possible to really phase well the HLA gene. So on the table, you can see again the 5 samples that we sequenced, order by read length. So it's pretty clear that the longer the read lengths are, the better the phasing is. But yeah, really for us, it's demonstrating that it's possible to phase a sample from FFPE DNA, which is a real advantage. So to conclude, this workflow can really open the door to a number of important applications. And yeah, the key message, I think it's important to understand that now FFPE can be successfully analyzed using HiFi sequencing. It's really the combination of the Covaris extraction and the PacBio library prep that provide this end-to-end workflow. And finally, long-read sequencing enable comprehensive variant detection and phasing from those FFPE samples. Thank you.